pleasure to introduce KETAN for his PhD defense today. KETAN has joined my lab a few years back. I can't remember exactly, but
2013.
2013. And his publication just came out last week, so it's fantastic. The timing couldn't be better. KETAN has done some really good work in my lab. He's taken on a project that was really difficult and really frustrating at many points. And it was all the way along, even for the review. And really, the basis is that what we do is subtle. Okay, we look at how cells move, and we look at why they move, faster or better direction, and everything we look at is subtle. And somehow, as scientists, we understand that biology is subtle, but as reviewer, it seems like people forget. They want something that is either black or white, no gray. 20% does not matter. An increase or decrease of 20% is not significant. And if you just think for a minute, if your core temperature decreased by 20%, you're dead. Okay? But all these people that are reviewing papers forget about that and assume that if it is not black or white, then it's not relevant or significant. And so I think that KETAN has been able to go despite that and really identify some highly significant biology and persevere through his PhD, and that's something that hopefully is going to help him for the rest of his career. So, without further ado.
Now the real introduction. Obviously, his mentor will give good introductions, but this is the real stuff.
Okay, well, thank you all for coming today to honor the life and his wonderful path that he's had through graduate school here with us.
And so, keeping in tradition with KETAN's philosophies, we are starting at 12:36. If it started any earlier, they would be like, whoa, is it KETAN's defense or what? So give it a half an hour or 5 hours, we'll start the presentation. Anyway, bye. Yeah. Anyway, but if you've met KETAN, you know that he operates on his own clock. And when he says, oh, I'm in the lab, it'll take me 20 minutes, you probably know that he's not in control. But otherwise, he's pretty good. So he is a man who needs no introduction. He was MCB president. He's been a part of all these social committees and all the nitty-gritties of MCB. But if you've ever met him for the first time, this is how he introduces himself. I'm not kidding. I'm Keaton, like Michael Keaton. And you're imagining Batman. No, someone like this, like Birdman. And you saw the first picture, like him in a tie, and he's here, he's dressed to the nines. This is what he looks like. But when we knew him, when we started to know him, he did not look like that. It was real effort. It was real effort to get him to look this good. And we'll show you how the progress was as he was going along his journey. But this is the final product, so don't be deceived. Just like his science, it has come along, it has matured, and now it's the final product. All right? So he starts off, eyes closed. We went through dozens of pictures trying to find the ones where his eyes are open. We found 5. And we gave up. Over here, he's on a ski slope. His eyes are closed. Kim tells him, please don't fall off the mountain. He finishes last on the slope. Hey, what took you so long? I was with the park ranger. Why? Fell off the mountain. So clearly, opening his eyes is something that he's learning. Slowly but surely, one day his eyes will open. And they're opening soon enough. And the same is true for his life partner. Look at his cat. A pair made in heaven. His eyes are closed, his eyes are closed. It's perfect. Whenever they're together, their eyes are closed, sleeping. And again, he wouldn't be with Kim if her eyes are open. No. Forget the smart scientist. No, eyes are closed, important. The big picture, But, you know, there's more. Hold on.
So eventually, I think he decided that he'd be like, all right, I'm wise to your game. You've all figured out that I have to keep my eyes closed. So he decided to hide it by just only wearing sunglasses all the time, wherever we went. And then he went through so many different pairs of sunglasses in 1 year. I don't even have numbers for this anymore. And eventually he figured out, well, this is not a sustainable trend. I need to figure it out. And so as he's maturing through grad school, he's also maturing and figuring out how to take it. So as you can see, he starts with one eye, tries the other eye, startled when he opens both his eyes. But then eventually it comes back to normal, right? Now we have the lovely individual that we have here today. And as he was going through this life-altering transition of opening his eyes, he also discovered many hobbies that he enjoyed throughout his graduate career. One of which is his love for food. And regardless of what he's doing or what he's wearing, he always eats as if it's his last meal on earth. And if you look at him, you'll wonder where the heck he's putting it all. And we think that his life partner is helping to carry the weight for him. But still, as he's going through his grad school trajectory, he's done other things, like he has a lot of outdoor activities that he likes to pursue in, such as jungle gym. And also, one of his other main loves is music. And so, as he's going through his day-to-day routine, he's always listening to music and coming up with new bands. And he'll call me and he'll tell me about his new band that he's discovered, and usually it's something obscure, some new hippie thing that's just come out. And then recently he told me about this new band that he just found. He loved it. He was so happy. If I had ever heard of them, happily this time I can say yes. But at the end of the day, we've all enjoyed our ride with you. This has been a fantastic time getting to know you over the years, and we couldn't be happier and more proud of you as you're finishing up today.
Glad to see you on stage now. All right. Well, who needs enemies, right?
Friends like us.
All right. Well, thank you all for coming. For those few of you who don't know me, my name is Keaton, and I've worked in the Alphandari lab for several years now. And today I'll be telling you a story about facial development and how a particular cell adhesion molecule, KETAN plays a role in this process. So the cranial neural crest, or CNC as I will refer to them throughout this presentation, are a versatile population of cells that give rise to the face. And they are found in all vertebrates, or all organisms with a backbone. And you can see an example, or 5 examples rather, of that here in blue. So here we have the contributions of the CNC, and what you can take away from this right away are all the number of different shapes and sizes of bones that the CNC give rise to. And as you can imagine, this is an immense responsibility, and there's often the time that things go awry. And in fact, 1 in every 300 births has some sort of anomaly of the face or head. And these can come in one of over 700 craniofacial syndromes. One of that is shown here in this individual. This individual doesn't show an obvious defect of the jaw, which is shown here in green. However, if you take a close look at his ear, however, there is a bit of a misfold. In more severe cases, you can get defects of the ear, but you can also get defects of the jaw. Now, these syndromes can occur in, let's say, 1 in every 50,000 births, but one syndrome that is, or one disorder that is more prevalent is clefting of the lip and clefting of the palate, which occurs in about 1 in 1,000 individuals. And you can imagine the economic and social burden
this imparts on the patient, not to mention their families. And so it's for this reason why we study the cranial neural crest development and more so their migration. So the cranial neural crest arise at the intersection of this neural tissue here, which is shown in blue, and this non-neural tissue, which is shown in white. Now, during the development of the nervous system, this tissue, which is shown in green, peels off from the superficial layer and migrates into either side of the embryo, and where it can give rise to a number of different tissue types, such as bone, cartilage, and muscle. So as not to trivialize this process, I wanted to show you what's happening in the actual embryo. And so if we have our embryo here on the left, and we peel off that superficial layer, that white layer, what we can see is something quite remarkable happening. And that is the CNC are migrating as segments, and they migrate into these structures, which are called the branchial arches, which is denoted by these arrowheads. To study the cranial neural crest, we use Xenopus laevis as our model system. And the reason why are for the following. So one is that we can play with hundreds, if not thousands, of embryos in a single day. And moreover, we can synchronize their development. And we can synchronize them by using in vitro fertilization. So yes, the male is reduced to basically just his testes. The embryo is incredibly resilient and malleable. So what that means is we can take CNC, put it on plastic, and watch them migrate underneath the microscope. We can also watch their migration in vivo. And the way that we can do that is by taking advantage of cell fate mapping. And what I mean is the fate of these cells here in early development, the fate of these are known. And so if we inject, let's say, this cell right here and allow the embryo to grow up, what we see is that one cell actually gave rise to a large majority of the neural crest, as you can see here by this nice segmentation. Another way that we can visualize in vivo CNC migration is through the use of in situ hybridization, which is basically a method that allows you to visualize gene expression. So, for example, here you have gene expression of a neural crest marker in blue or purple. And what you're taking a look at here are embryos through the developmental timeline. And here at the dorsal aspect of the embryo are the CNC cells, and they're migrating at this time as a collective cell. But through time, they segment, as we just saw, and they enter these branchial arches, which are specifically known as the mandibular hyoid and then third and fourth branchial arch. I want to really drive home this point of the power of our system, not to mention just the awesomeness of this migration. And so here I have this movie for you. This is an embryo with the head positioned towards the left. Here in red, you can see the neural crest, and this leftmost stream is already the mandibular stream of cells that has migrated, or mostly migrated. And what you'll see happening is that the hyoid and then third and fourth CNC streams are coming down into the embryo. Now, we can take a look at CNC migration as a population, but we can also go one step further, and we can take a look at what single cells are doing. And so here you're taking a look at a neural crest cell that has been injected with an actin fused to an RFP, or red fluorescent protein. And those will appear as basically red lines. And we've also injected a GFP to basically outline the cell body. And if I play this movie, what you are actually seeing are a number of different processes happening at once. For example, firstly, we will see the cell sensing which way it would like to go. And then once it sends out its fingers and attaches to a substrate, it contracts, and you can see along this back edge that it can lift up its rear, severing those attachments. And so, as you can imagine, there are a number of different proteins on the cell surface that are helping to facilitate this process. And our favorite group of molecules is the ATOM family of proteases. ATOM is an acronym that stands for a disintegrant and metalloprotease. Basically, it helps describe two components of the molecule. There are also other components that comprise the molecule. There's a prodomain at its end terminus, and this prodomain helps the protein fold, as well as prevents premature cleavage of substrates. Following that is the metalloprotease domain, or the molecular scissors. Following that are two domains that help in protein-protein interactions. Following that is an EGF-like domain, followed by a transmembrane domain, and then at the C-terminal end is a cytoplasmic domain. And one atom that we care immensely about in our lab is atom 13. And the reason why is because it is expressed in the CNC, as you can see in this picture of it in Situ Hybridization. And moreover, it is important for CNC migration. So if we inject CNC cells with this green fluorescent protein, or a catalytically inactive atom 13, and then translate those cells into wild-type embryos, what you can see for controls is, again, nice segmentation, but with the catalytically dead atom 13, you can see that the only stream that migrates is the mandibular stream. And I should also mention that atom 13s are cleaving, or can cleave, a number of different cell adhesion molecules. And my favorite cell adhesion molecule is cadherin 11. Cadherin 11 is a lot more simpler in construction. It's composed of 5 cadherin repeats, followed by a transmembrane domain, and then finally a cytoplasmic tail, which effectively connects it to the actin cytoskeleton via these catenin molecules. Now, during the lifetime of cadherin 11, it can bind in cis, but more predominantly binds in trans, and it does so through the use of its distalmost cadherin domain. Also during its lifetime, when atom 13 comes in close proximity to it, atom 13 can actually cleave the molecule between its third and fourth cadherin domain, releasing this fragment. Now, a previous student in our lab saw this and was curious as to whether this fragment was actually facilitating migration in any way. And so she played with the overexpression of the full-length cadherin 11, the fragment, as well as a tool called a morpholino, which helps in knocking down a protein. And she injected those different mRNA and morpholino into this CNC mother cell, allowed the embryos to grow, and later scored the embryos for migration or no migration. And if we go to her data, we can see here in her controls, nice segmentation of the CNC, and in embryos where she injected a triple morpholino, so a morpholino that knocks down atom 9, 13, and 19, that the neural crests were stuck along the dorsal aspect. However, when she overexpressed the fragment, she got a partial rescue of migration. And if we go to this graph here, where we're taking a look at CNC migration on the y-axis, and we set our controls to 100%, we can see that the embryos that were injected with the morpholino do in fact have reduced CNC migration, and moreover, those expressing the fragment can rescue this decrease. Now, in a similar experiment, she left the metalloproteases alone, and then played with the levels of full-length cadherin 11. And when she overexpressed this cadherin, she saw that, again, the CNC were stuck along the dorsal aspect. However, when she overexpressed the full length plus the fragment, she could get a partial rescue, as shown again in this graph. And she left us off with this question that's been the driving force of my thesis, and that is, how is EC123 promoting CNC migration? Now, there are two hypotheses that we put forth to answer this. And the first has EC1 working through a de-adhesion process, in which it works downstream of atom 13 to disconnect cells by preventing or inhibiting the homophilic binding between cadherin molecules. So to test this possibility, another student in our lab, Genevieve, took advantage of the fact that homophilic binding relies on residues in the EC1 domain, and what she did is she mutated those domains, those residues. And she created this construct, non-adhesive EC123, or NNA EC123, and then took that construct and basically repeated Kate's assay. And here, we're taking a look at inhibition of CNC migration on the x-axis. With our control set to 0, we can see that embryos overexpressing cadherin 11 do in fact show inhibition. And similarly, if we, similar to Kate's data, if we overexpress the fragment, we get a rescue. Now, with the non-adhesive EC123, we get rescue. So this suggests that this first hypothesis is not quite the explanation for the biological activity of EC123, and rather that it's acting through signaling receptors, which brings us to my model and my model in which we think that EC123 is interacting with a cell surface receptor, which initiates a signaling cascade that are both important for cell migration. Now, to start my project, we focused on these two molecules here. These kinases have already been implicated in cell motility and are hubs of signaling. And so we could take a look at the phosphorylated state of these proteins in embryos overexpressing the fragment and see if they're elevated. And what we see is that phosphorylation of AKT is in fact significantly elevated. However, phosphorylation of MAP kinase is not. Now, if EC123 is acting as a ligand, you might want to see more directly its effect. And so one way that we can do that is by using or by producing a recombinant EC123. And so we took E. coli, expressed our fragment, and purified it from the E. coli. And now we have it in hand, and we can play with the exposure time as well as the concentration. And so we took a wild-type CNC and treated it for just 5 minutes with our recombinant. And what you see is something very similar to the previous slide, in that phospho-AKT appears to be increasing. And if we go to the numbers, it does, in fact, significantly increase. And similar to the last slide, phospho-MAP kinase doesn't appear to be changing very much. Now, if you're as paranoid as I am about your data, you are thinking, oh no, there are contaminants in the purification, and those contaminants are somehow eliciting this effect. So what you would do if you were me is you would run a set of controls which would require you to produce a specific antibody to your fragment so that you could deplete it from your treatment solution and use that solution to then treat your CNC and perform a Western blot. And what you can see in our depleted solution is that phospho-AKT is in fact brought down, and in our control of controls, we can see that AKT our phospho-AKT levels still rise. So with my paranoia at a minimum, I could then move forward and assay whether or not this fragment can promote migration in cells, similar to what Kate saw earlier with the other fragment. So we go to our in vitro assay, allow the cells to move in the presence of our recombinant, and this is an example of what we might see. And what we can do with this data using cell tracking data, using cell tracking algorithms, is actually pull out different parameters of cell migration. For example, there's persistence. Persistence is defined as the net displacement of the cell divided by the total displacement, which basically, persistence is basically a measure for its directionality. How often does a cell go in a particular direction? So a value of 1 would mean that it is a straight arrow. And actually, cells treated with a recombinant do have a significantly higher persistence. Now, when we took a look at velocity, however, there's no significant change. All right, so this is what we see so far. EC123 can initiate AKT signaling, but we still don't know if AKT is important for CNC cell migration. And so one way that we can test this possibility is by taking advantage of the fact that PI3 kinase is important for AKT phosphorylation, and moreover, this inhibitor is well known to block PI3 kinase. So we can take this inhibitor, go back to our in vitro assay, and treat our cells with the inhibitor, and we would see something like this. And what we can do with this data is we can stop the movie at different time points as such, and what we can see qualitatively is there already seems to be an effect. Now, to get some numbers out of this, what we can do is trace the explant and then basically compare the different explant areas. And if we take a look at the numbers, we can see that for the duration of our experiment, the inhibitor does significantly reduce migration. Now, this is great, but we want to go in vivo, right? The real power is in vivo. And so we treated embryos with the inhibitor, and what you're taking a look at here is again in situ hybridization for a CNC marker. And just to describe how we quantify the CNC migration, for the mandibular arch, we use the top of the eye down to the ventralmost point, and for the remaining 3 arches, we use this topmost point to this ventral point, and normalize it to the head height. Now, before I show you the data, I just wanted to point out that arch 3 and 4 already appear to be affected. And if we actually go to the numbers, we can see that across the board, CNC migration is affected. Now, to make sure that this inhibitor is working as expected, we could take a look at the phosphorylation state of AKT. And when we do that, we can see embryos treated with the inhibitor do in fact have a reduced phosphorylated AKT. All right, so our working model is that EC123 signals through AKT to promote CNC migration. Now, what's left to be investigated is which receptor is mediating this process. And to help us in this, we could use 2 criteria to help us narrow down our candidates. One, it's very obvious, it's got to be upstream of PI3 kinase AKT signaling, and the second is that it should be expressed in the CNC. And so that brought us to these 6 receptors. These 4 are from the same family, the ERB family, and here we have PDGFR alpha and FGFR1. So to begin, we just simply asked, can the receptor bind to the fragment? And we assayed this using co-immunoprecipitations, and what we see is that all 6 of these receptors can bind to the fragment. Now, I know that this can be a little bit disconcerting, so as a control, we co-transfected the fragment with these flag-tagged cytosolic proteins, and we don't see any interaction, so that was good. Now, if you remember from earlier, we have this construct, this non-adhesive EC123, and it can promote migration. And we might expect that this fragment can bind to the receptors, and when we take a look, very similar to the wild-type fragment, it also can bind to all 6 of our candidates. Okay, so how do we narrow down which of these is upstream of PI3 kinase AKT signaling? Well, we can use this assay here, where we transfect the receptors, starve the cells of other signaling molecules, treat it with our recombinant, and then finally extract the cells and do a Western blot for phosphorylation of AKT. And what you see is a general trend, a downward trend in phosphorylation of AKT. However, very clearly, there is one receptor that jumps out, and that's this ERB2 receptor. To ensure that it's specifically this ERB2 receptor, we can use a potent inhibitor called mubritinib and treat our cells with mubritinib prior to actually treating them with our recombinant. And what we see in our controls is that phosphorylation of AKT does still climb. However, that is abrogated by introducing the cells to mubritinib. Now, what's so peculiar about this data is that ERB2 cannot homodimerize with itself, and in fact, it requires the binding to one of the 3 other family members, which means that the overexpressed ERB2 from our last slide was most likely binding to endogenous receptors in the 293 T cells, and we actually have mass spec data to support this idea. The other thing that I wanted to mention is that these receptors, by default, are in a closed or inactive conformation. However, upon binding of their canonical ligand, they can adopt the active conformation and bind to ERB2. After this, a number of phosphorylation events occurs in its cytoplasmic domain and allows PI3 kinase to dock at the cell membrane where it can initiate AKT signaling. And I gotta say that we tested the different permutations of these heterodimers, and ERB2, ERB3 actually could still elevate phosphorylation of AKT in the presence of a recombinant. So we wanted to take a look at the receptors just to make sure that we were on the right track. So we looked at, the gene expression of the receptors in the CNC, and what you're taking a look at here is that ERB2, 3, and 4 are all enriched in the CNC versus the embryo. And so that left us with these 2 candidates. Now, PI3 kinase docks to a particular sequence, and that sequence is defined by this YXXM motif. And as you can see here, there's 6 of these putative binding sites in ERB3, but not ERB4. So we focus our energy on ERB3. And we did a number of co-immunoprecipitations with PI3 kinase as well as this ERB3 receptor. And here's that data. So you can see that association of PI3 kinase with the receptor is in fact elevated in cells treated with our recombinant. And that association jumps up immensely when you co-transfect ERB2 with ERB3 in these cells. And the association is further elevated when you treat these cells with our recombinant, and that is a significantly increased association. So our working model at this point is that EC123 binds to ERB2, ERB3, and initiates signaling through PI3 kinase and AKT, all of which is important for CNC migration. Now, this model would suggest that ERB2 is important for AKT phosphorylation in vivo, and moreover, that this receptor is important for CNC migration. And so, to see if this was the case, we used a morpholino of ERB2, so that morpholino, remember, brings down protein expression. And as our first experiment, we wanted to make sure that the morpholino was doing its job. So we injected embryos with a stabilized ERB2 construct, which you can see here by Western blot. And we also injected embryos with the morpholino with this stabilized construct, and you can see that it doesn't allow translation of this transcript. Okay, so with our morpholino working, we then checked phospho-AKT and CNC injected with the morpholino, and as you can see, there's a dramatic decrease in phospho-AKT. All right, with that in order, we then went and took a look at CNC migration. Using our morpholino, here are our controls. We can see nice segmentation in the controls. In embryos injected with the morpholino, you can see that there's a reduction in CNC migration. And in embryos injected with the morpholino and a non-targetable ERB2, it appears that we can rescue that migration. And if we go to the numbers, we can see a significant drop-off in CNC migration in embryos injected with the morpholino. And we can get a significant rescue in embryos injected with the morpholino plus the non-targetable ERB2. So one thing that I must mention, though, and that maybe some of you have already noticed, is that the pattern of migration is still occurring. So the CNC is still entering the four arches, but it appears that the number of CNC cells seems to be decreased. And so we were a little bit concerned that the morpholino was actually hitting the CNC prior to their migration. So to test this, we again injected embryos with the morpholino and stained them for CNC markers, but stopped their development prior to migration. So for example, here you have an embryo that was injected with RFP just on this right side. And here you have an embryo injected with morpholino on this right side. And as you can clearly see, there's a reduction in CNC gene expression. And so we need to somehow inhibit ERB2 function just prior to migration. So one way we can do that is by using the inhibitor that we used earlier. And I've also thrown in, for good measure, a pan-ERB inhibitor. And we can treat embryos with this. And when we did, we were a little bit surprised to not see any effect. Now, if they were actually having an effect, they should hypothetically reduce phosphorylation of AKT. And when we take a look at that via Western blot, they're not doing that. And so we thought that there was an issue with penetrance. And so we went back to our in vitro assay and treated CNC with the two inhibitors. And what you can see is the following. Mubritinib, or the inhibitor to ERB2, does have a slight effect on CNC migration, and then Canertinib has an immense negative effect on CNC migration. And if you go to the numbers, you can see that these are significant trends throughout the duration of this experiment. Moreover, you can see that inhibiting just ERB2, inhibiting ERB2 is not as potent as inhibiting all four receptors, which suggests that EGF1, ERB1, ERB3, and ERB4 are contributing to CNC migration. And what you also see is that inhibition of PI3 kinase is similar to inhibition of the ERB2 receptor, again, arguing for a case that ERB2 is connected to AKT signaling. So our current working model is that EC123 binds ERB2, which initiates a signaling cascade of PI3 kinase and AKT, all of which are important for CNC migration. Now, as I mentioned just a second ago, it appears that the other ERB family members are important for CNC migration as well. Now, I just wanted to emphasize that this is the first time that we have seen pro-migratory activity, the first time that anybody has seen pro-migratory activity by a cadherin fragment in an animal. So how does this compare with other models? Well, in the case of E-cadherin, atom 10 and atom 15 can cleave the molecule very similar to what we've seen with cadherin 11. And this extracellular domain can actually bind ERB2 and ERB3 as well, and initiate signaling through MAP kinase and AKT, which promotes cancer cell migration. And this parallel between cancer cell migration and the neural crest is just one of many that are growing. And so it's really tempting to say that the neural crest can be used as a model system for cancer metastasis. So in the future, I would love to take a look at the residues facilitating the interaction between EC123 and ERB2. And I would also like to see if this fragment is somehow stabilizing canonical ligand binding, canonical ligand receptor binding. I would also like to take a look at the other cadherin ectodomains and the other proto-cadherin ectodomains to see if they have signaling potential as well. And as I mentioned, there are other receptors and signaling pathways that would be worth investigating in CNC migration. So there are plenty of people to acknowledge. Dom, thank you so much for taking me in and making me the scientist that I am today. I am often just floored by your willingness to adapt new methodologies to answer any question. And I gotta say that, as you mentioned in the introduction, detailing mechanisms in an organism is incredibly difficult, and the fact that you've been doing this for so long is just wild. Alain, thank you so much for teaching me everything that I know about embryology and drawing me with probably like 100 pictures of embryos. I must thank the Alfandari Lab past and present. Thank you especially to Vikram, Vanessa, Genevieve for all your support. My committee members, Craig, thank you so much for being a champion of subtleties. Honestly, just as Dom said, I thought I was dead in the water a few times, but you said, get some numbers and see what happens. And so I must thank you for that. Raphael, thank you so much for making sure I had a keen eye on my inhibitors. There was a time that I didn't, and it set me back a little bit. Scott, thank you for being a champion of graduate students. I'm not the first person, and I'm not the last person to say that, and I'm not just saying that because we're about to go into a closed session. So thank you very much. Dr. Gross, thank you so much for being a champion of graduate students and undergraduate students. I hope that I can achieve as much success as an instructor as you do. I also must thank my former mentors, Craig, my former undergraduate mentor who passed away a few years ago, and Brandon and Nicole must thank you. The supporting cast, without which the world would not turn. Thank you to Sarah, Carrie, Jamie, Bob Savola, Scott. Of course, you got to thank the money, right? Thank you CBI for bringing me together with so many different disciplines and allowing me to meet so many different people. To Baskey, thank you. I can't even remember how many different Baskey faculty I've spoken to in the last 6 months as I try to transition out of graduate school. And of course, thank you to the MCB program. Thank you to the old guard, so Leslie, Jody, Ralphie, Ankit, Russ, Nick Reiner. Thank you all for taking me in. Of course, I must thank my family. Thank you to my sisters for dealing with me growing up. Thank you to all of them for your love and support. Even if I didn't become the doctor that can treat it. Thank you so much for giving me the tools to succeed. And I also must thank my second family here at UMass. In no particular order, I've got these crazy kids. I must thank my lab cousins, Jesse, Gabe, Chelsea, Agnes. Thank you guys for just being yourselves day in and day out. You guys crack me up. And honestly, thank you for making my life so rich and full of laughter. You know, it's a funny thing to be hurling through space on this beautiful spaceship we call Earth. And I'm so blessed to be to share it with you. I must also thank my friends from back home, and these two stand-up guys here. And of course, I must thank my fiance. Thank you so much for waiting patiently. You're a constant source of inspiration, and I can't wait to be by your side again. And with that, thank you. Now it's If you're ever wondering what is happening out of the laboratory, this is what is happening.
Great job. Fantastic to see it all come together. So at the end, you had an idea that perhaps earlier events were being impacted. Those in vivo experiments, however, weren't totally conclusive, but your in vitro results seem to suggest it's a pretty cell autonomous effect on cranial neural crest. But if you did have the next step experiment to do, I mean, do you still wonder whether earlier events are in fact contributing, you know, to the overall the extent of the phenotype, the extent of movement of cranial neural crest, or is it just really in the neural crest?
Good question. I mean, I would place my bets on that. It's more so the downstream events, because a number of different. I'm sure you're familiar with the EMT. A number of different cellular changes that are occurring between, you know, specifying or inducing the CNC and actually having them migrate. So I think that the receptor, the ERB2 receptor, is important for both events, but I'm sorry, I'm going off here.
So for instance, the earlier events of nervous system development that create the brain, and then you have neural crest come out. If you're not having robust convergent extension and movement during the earlier stages of gastrulation that will then produce that early event, I can imagine that impacting, you know, downstream the effects you see on the pharyngeal arches.
Oh, I see. So.
And so are the impacts to cadherins through your system affecting those earlier events?
Oh, I see. Yeah, it's quite possible. Actually, yeah, it's quite possible. When I've done a in situ hybridization in embryos that are overexpressing the fragment, for example, there seems to be a proliferation of the CNC population as a whole. So prior to migration, they already seem expanded. So the fragment, yeah, it could be having an effect prior to migration. Does that answer your question?
Yeah.
All right. Everybody is asleep.
I have a question. So in the animals that you treat, and you see the subtleties in the change in the pharyngeal arches, do they ever become adults? Do you see what happens in like later forms of life?
I have not taken a look at the later stages. Thanks.
I missed your name.
Kathleen?
Kathleen.
Our current, yes. Our
So
current.
should we recommend him to go into teaching?
Oh, I think he's thought about teaching. I think he knows that he's excellent and he's discussed it. He'll be wonderful. But, sometimes he might want the combination of teaching and research. So I'm not sure what his plan is next. But he'll be an unusually good teacher, like maybe extremely good. That was the first time I saw that his undergraduate must have been at University of Florida with Lou Gillette. Did you know? He thanked his previous mentor and it said Lou Gillette. So I know that person. Now I understand a little more about Keaton, which I always called him KETAN. He never corrected me. And only today I learned his name is, you say Keaton. How do you say it?
In Hindi we say Ketan.
Oh, I always say, hi KETAN. So I didn't know. So I think he's considering teaching. But he may want a few more years of doing research in a lab, and then with that he'll get a better, if he wants to teach at the university, he'll get a better job. So I'm not sure what his plans are. But yeah, you see, and this is like such a nervous day, and he's still great, right? He was still very great. And his slides were really excellent, like, the clarity and all. So he was teaching in the class, he was teaching students how to work with cells, how to be, he had to even teach them how to sit and how to work, and because everything you do affects, how it turns out. And he was great. So yeah, I think he'll be very good, exceptional, and motivational to many people.
I did my PhD in engineering at UM.
Oh, okay. What's your area of engineering?
Mechanical engineering, and now I work at nuclear plants with Florida Power Plant.
Oh, okay, neat. My stepson went to mechanical engineering here at UMass. So yeah, but nice. Well, I may see you later. So now we have to leave, and what they do is they question him, they grill him for an hour or so, and then there's a